So if you're joining us today, you're probably here because you like to stay on top of, the big scientific breakthroughs, but without getting totally bogged down in all the dense medical jargon.
Right, which is exactly what we're going to try to do here.
Exactly. And for this deep dive, we are looking at something pretty incredible. I mean, it's a May 2026 article from Science Daily, and it covers a massive discovery out of the Indiana University School of Medicine.
Yeah, and it's definitely a fascinating one.
It really is. Because the goal today is to look at how scientists haven't just found like another way to clear Alzheimer's plaques, but they've actually found a potential way to help the brain actively fight back.
Which is a huge paradigm shift. I mean, this isn't just about what they discovered. It's really about why this could fundamentally change how we treat neurodegeneration altogether.
Totally. It's almost like a scientific detective story.
Right here.
But to really understand why this Indiana University discovery is such a big deal, we probably need to establish how Alzheimer's is currently treated, right?
Sure. Yeah, that's crucial context. So recently, the FDA approved a couple of disease-modifying drugs. You've probably heard of them, lecanemab and donanemab.
Right. Those have been in the news everywhere lately.
Exactly. And they do represent a milestone. They work basically by clearing out amyloid plaque buildup in the brain.
Which is the sticky stuff that builds up between the cells.
Yes, exactly. And clearing that plaque helps stabilize patients. It slows down their further cognitive decline. But the new research we're looking at today targets something completely different, an enzyme called IDLE.
Okay, let's unpack this because I was thinking about this earlier, and tell me if this analogy works.
Sure, let's hear it.
If current drugs, like lecanemab and donanemab, are basically like bailing water out of a sinking boat to stop it from going completely under, then targeting this IDLE enzyme sounds more like a strategy to actually, I don't know, patch the hull and make the wood water resistant.
I actually love that analogy. It's very accurate because bailing the water is necessary, but it doesn't fix the leak.
Right, you're still in a leaky boat.
Exactly. And targeting enzymes like IDLE offers a very specific advantage. Professor Kim, one of the lead researchers, explained this perfectly. Enzymes have these really well-defined active sites, which scientists call pockets.
Pockets, like literally just spaces where things fit.
Pretty much. They are highly specific three-dimensional shapes, and drugs can be designed to attach directly to these pockets to block the enzyme's activity.
Oh, wow, so it's super precise.
Very precise. It means you can hit the exact right target with minimal side effects. You aren't just broadly altering the brain, you are, competitively inhibiting one specific machine.
Okay, that makes sense.
Yeah.
But knowing how to target an enzyme is one thing. Figuring out where to do it in the brain led scientists to a completely unexpected discovery.
The plot twist, basically.
Right, because here's where it gets really interesting. So, researchers created two different animal models of Alzheimer's, right?
Transgenic mice, yeah.
And they deleted the IDLE gene in two different types of brain cells.
Right.
In one model, they deleted it in the microglia, the immune cells, and in the other, they deleted it in the neurons.
And based on everything we knew about brain pathology, the scientists fully expected the microglia to be the star of the show.
Right, because wait, if the microglia immune cells are the ones actually producing the IDLE, and they're basically the brain's janitors cleaning up the mess, shouldn't altering them have the biggest impact? Why did the neurons steal the show?
What's fascinating here is that this perfectly illustrates why you have to test these biological assumptions. Because it seems like simple logic, right? If the janitor makes the enzyme, fix the janitor.
Exactly. It seems so obvious.
But the results completely contradicted that. Removing IDLE from the microglia had barely any effect on the disease pathology.
Which is wild.
It really is. The most striking systemic effects only happened when they removed IDLE from the neurons themselves. Removing it from the neurons didn't just passively stop the plaque, it actively changed the entire cellular environment.
So the neurons aren't just sitting there being attacked.
No, not at all. They are actively regulating their own microenvironment.
Okay, so because the neurons reacted so powerfully, the researchers obviously looked closer, and they realized the benefits went far beyond just plaque removal, right? Like they were actually rewiring the brain's defenses.
Yes. This is where we get into the really heavy physiological changes. Deleting IDLE in the neurons didn't just significantly reduce the amyloid plaques, it also reduced the levels of APOE.
And APOE, specifically APOE4, is the big one, right? Like the genetic risk factor.
It is the single greatest genetic risk factor for late-onset Alzheimer's.
Wow. Okay, so it dropped the APOE level.
You're right. And it increased specific receptors that regulate both APOE and those amyloid plaques. This helps maintain communication between neurons and supports healthy lipid metabolism.
Lipid metabolism, so like the processing of fats in the brain.
Exactly. The brain is incredibly rich in lipids. Fats and cholesterol are what build the cell membranes and keep synapses functioning. But they need APOE to transport them.
Oh, I see. And APOE4 is just really bad at that job.
It's terrible at it. It's structurally unstable, it doesn't deliver the lipids, and it actually encourages plaque to aggregate. So by deleting idle, they fix this broken transport system.
It's like realizing your computer has a virus. You don't just want to delete the corrupted files, the plaques. You want to actually upgrade the firewall and improve the processing speed, which would be the lipid metabolism and the cellular communication.
That's spot on. And if we connect this to the bigger picture, it speaks directly to a point made by Dr. Hande Karhan, another key researcher on the team.
Right. What does she say about the diagnosis timeline?
She noted that patients are almost always diagnosed late. By the time someone is showing cognitive symptoms, that plaque accumulation is already substantial.
The damage is done, essentially.
Exactly. So she argues that we need to decrease amyloid, yes, but we also urgently need to increase resilience.
Because if a patient already has severe plaque buildup, just clearing it isn't enough to save them.
Exactly. Enhancing those neuroprotective effects, restoring the lipid metabolism to resist further cognitive decline, that is what could actually maximize clinical benefits.
Okay, so they've got this incredible dual action mechanism in these animal models. It clears plaque and it builds resilience. But, with these incredible results in the lab, what are the actual next steps to turn this into a drug you or I might find in a pharmacy?
Well, the translational science phase is starting now. The team is exploring approaches to develop actual drugs that target that idle enzyme pocket we talked about earlier.
Small molecule drugs, right?
Right. They need compounds that can cross the blood-brain barrier, find the idle enzyme, and bind to it without causing other issues. So future studies will heavily focus on testing the safety of these potential compounds.
Right, making sure it doesn't just like braid something else in the body.
Exactly. And then evaluating their efficacy in preclinical models. But there's another aspect to their future plans that is incredibly important.
Yeah, that's it.
They also plan to investigate if blocking idle preserves synaptic connections and reduces tau pathology.
Wait, so what does this all mean? Why are they looking at tau pathology now when this whole conversation has been about amyloid plaques and APOE?
This raises an important question, and it really gets to the heart of how tricky Alzheimer's is. It is essentially a multi-headed beast.
Right.
Tackling amyloid plaques and APOE is a huge victory, but tau pathology is the other major hallmark of the disease.
So what exactly is tau, just for anyone who might not know?
Sure. So inside the neuron, you have microtubules. Think of them like the internal scaffolding or like railroad tracks that transport materials inside the cell.
Okay, railroad tracks inside the cell. Got it.
The tau protein normally acts like the cross ties holding those tracks together, but in Alzheimer's, they detach, they clump together into tangles, and those railroad tracks completely fall apart.
Oh, wow. So the cell just sort of collapses from the inside.
Yes, the neuron dies. And tau tangles are very closely correlated with severe memory loss. So the fact that targeting this one enzyme idle might also impact tau.
That would mean it's hitting all the major targets at once.
Exactly. It represents a unified approach to a highly complex disease. You're fixing the outside of the cell and potentially saving the inside structural integrity, too.
That is, that's wild. It's so hopeful. So to kind of wrap this up for you listening, think about the journey we've just covered here.
It's quite a shift from where science was even a few years ago.
Totally. We went from the limitations of current plaque-clearing drugs, just bailing out the boat, to this massive biological plot twist where neurons are actually the key players, not just the immune cells.
Right.
And now, by targeting these specific pockets in the idle enzyme, scientists might be able to create highly precise treatments that don't just clear the garbage, but actually build up the brain's structural resilience.
Which is really what patients need.
Exactly. And this matters so much. Whether you are tracking medical science because of a loved one or you're just, fascinated by human biology, this research shows that we are moving from merely slowing Alzheimer's to potentially teaching the brain to shield itself.
Yeah, and actually I want to leave the listener with a final thought to really mull over, based purely on what the researchers are planning next.
Oh, definitely. Lay it on us.
The source mentions that future research will look into whether blocking idle can also reduce that tau pathology and preserve the synaptic connection.
Right, the inside the cell collapse.
Exactly. So if manipulating the single enzyme can simultaneously reduce amyloid plaques, suppress the dangerous APOA4 protein, boost lipid metabolism, preserve synapses, and potentially stop tau pathology,
Hmm.
are scientists no longer just looking at a trigger for Alzheimer's, but rather an undiscovered master switch for brain aging itself?
Oh, wow. A master switch for brain aging. That's something to think about. It really makes you wonder what else our cells are secretly capable of. Keep digging deeper, everyone. We'll catch you on the next deep dive.